Welcome to the future, where we see flying cars, AI taking over the world, and mice. Sorry, what I meant to say was gene editing. While editing our DNA may sound like a futuristic concept, technologies are advancing so rapidly that the potential to edit human genes may not be as far-fetched as we believe. So, why were the 3 blind mice blind anyway? It's possible that they suffer from a genetic disease like BCD, as we haven't found a cure for them for hundreds of years. Luckily, the future is looking brighter than ever for curing genetic diseases. Welcome to the genetic basis of stuff and things. Alright, little mouse, let's see what we're working with here. Progressive night blindness, constriction of the visual field, and decreased visual acuity. These are symptoms of Bietti crystalline corneoretinal dystrophy. BCD is an autosomal recessively inherited genetic disease that affects the choroid, retina, and cornea of the eye and is characterized by yellow-white glittery crystalline deposits throughout the posterior pole of the retina. The CYP4V2 gene is the only gene associated with BCD. Recent studies have demonstrated this as the causative gene of the disease, with 80% of BCD mutations occurring in exons 7 to 11. The gene encodes an enzyme involved in oxidizing omega-3 polyunsaturated fatty acids in the eye. Lipids in the optic discs are metabolized in retinal pigment epithelial cells to maintain the function of photoreceptors and prevent the buildup of fatty acids in the eye. Any single mutation in the mutation hotspot of the CYP4V2 gene can result in a faulty enzyme, causing an accumulation of intracellular fatty acid in the eye, which, over time, form the crystal deposits in the retina. Sadly, there is no treatment available for BCD, but nucleic acid-based therapies have the potential to cure genetic diseases, and a lot of research is already being undertaken to further our understanding of genetic diseases. Researchers from Peking University in Beijing, China, worked on developing a CRISPR-mediated treatment for BCD, with the resulting paper published in 2024. Firstly, the researchers generated a novel humanized knock-in mutant mouse using CRISPR. The mutations resulted in the deletion of exon 7 of the CYP4V2 mouse gene, and the researchers found scattered yellow-white crystalline deposits in the mouse after 6 months, confirming the BCD mutant mouse model. They then used a CRISPR-Cas9 method combined with homology-independent targeted integration to restore the CYP4V2 gene. Firstly, the Cas9 protein is guided by a single guide RNA, which generates a double-stranded cleavage on intron 6 of the CYP4V2 gene. Then, a HITI donor inserts the restored sequence and a stop codon into the DNA break, achieving precise integration and restoring normal function of the gene. They did this because most BCD mutations occur in exons 7 to 11, so by inserting the correct sequence with a stop codon before this area, exons 7 to 11 that may contain fault are truncated and normal function of the enzyme can occur. The researchers found that the HITI-based method restored visual function in the mutant mice. 11 months after treatment, a follow-up session investigated changes in the mice's retinal pigment epithelial cells and photoreceptors, finding a complete and sustained recovery. While it is safe to say that BCD is a debilitating genetic disease, who knows what the future holds? The researchers who cured the mice even go so far to suggest that one single injection in BCD patients could achieve lifelong effectiveness against BCD. Considering the trajectory and rate of current research on gene therapy, the idea of curing heritable diseases in humans is becoming a more palpable possibility by the day. But hey, That's just a theory, a gene theory. Thanks for watching.